morning and good afternoon, depending on what coast you are on. I'm happy to be opening up the second Artificial Intelligence and Machine Learning seminar being hosted out of NASA Ames from the Exobiology branch. This is essentially a part of an AIMO initiative between myself and Caleb Sharp as part of my NPMP project. And today I'm very happy to be presenting Anirudh Prabhu and Wong from the Carnegie Institute for Science. They will be presenting on their work involving AI/ML that's actually looking at machine learning and laboratory work to work towards identifying biogenicity from both present and past specimens. So Mike and Anira, and just real quick, we're going to save questions until both speakers have gone at the end. So if you have questions, to save them or put them in the chat and we'll answer them at the end. So Mike and Anirudh, please take it away.
Okie dokie. So I'm going to attempt to share my screen again, and you let me know if this is successful. So here we go. Hopefully you're seeing my slides, and hopefully it's now full screen. Can I get a verbal confirmation about that? Because I can't see any of it. Okay, great.
That's
awesome.
Perfect.
Super. And if anything ever breaks, if my slides aren't moving, you can't hear me anymore, just yell at me. I know sometimes the internet has a mind of its own. So yeah, let's get started. So it's an honor to be here at the AI/ML Astrobiology Seminar Series. I'm Mike Wong, as Ryan said. I'm a NASA Sagan Fellow working at Carnegie Science, and I'll be co-presenting this project today with my good friend and colleague, Dr. Anirudh Prabhu, who's a research scientist and PI at Carnegie Science. I'm a planetary scientist and astrobiologist by training, and Anirudh is a data scientist by training with expertise in developing and applying ML and AI to all kinds of astro, geo, biochemical data sets. So it's been a really fruitful collaboration working with him over the past 2.5 years. And we're so happy to be here to share with you some of the most exciting results that we have. We'll be talking about searching for life in the solar system using machine learning and pyrolysis, gas chromatography, mass spectrometry. I'll be giving the first half of the talk as an astrobiologist. I'm going to be speaking about biosignature detection in general, just introducing that to everybody, motivating our proposed biosignature technique, and introducing the instrumentation that we're going to use. And then Anirudh will take it from there and talk about our data processing pipeline, our machine learning methods, the results and implications of our work. So let's start off by giving you a taste of how astrobiologists think about life detection. I was lucky enough to be on the scientific organizing committee for the Biosignature Standards of Evidence Community Workshop held in the summer of 2021. This was a joint effort between NASA's Network for Life Detection, or NFOLD, and NASA's Nexus for Exoplanet System Science, Nexus. The idea of this workshop basically was that we need to agree upon how we're going to identify, interpret, and report potential biosignature findings to the rest of the scientific community and to the general public. And the attendees produced a white paper that was open for community input and is now published on the archive. You can follow the link there to the final paper. It's 86 pages long, so that's quite a read. In case you don't want to wade through all of it, I've got one slide summarizing it for you. So the framework for life detection follows 5 key questions. Number 1, have you detected an authentic signal? 2, have you adequately identified the signal? 3, are there abiotic sources of the signal? 4, is the signal consistent with expressions of life? And 5, are there independent lines of evidence that can help us raise our credence in having detected life or lower our credence in that detection? So because all these questions are highly interrelated, we sort of group them schematically like this, where questions 1 and 2 feed back on one another to understand the detection, basically asking whether or not we've found something of interest. And questions 3, 4, and 5 do the same thing to help us understand the interpretation of that signal. In many cases, this is to try to understand the environmental context of the signal to tell if it is the result of biology or perhaps some kind of geochemistry. And then finally, question 5 is very critical because science is always an iterative process. From our discoveries, we should come up with new hypotheses that can then be sought to be tested to raise our level of confidence or lower our level of confidence in the detection of alien life. I want to make a note that this is not necessarily a linear kind of process. A modeler, for instance, might start by analyzing questions 3 and 4 so that when future observations are made through questions 1 and 2, we'll know what to make of those signals. Okay, so let's look at a simple case study to see this framework in action. Let's review the evidence for and against the detection of alien life in the Martian meteorite ALH 84001. So this is a meteorite that came from Mars, crashed on Earth, and was analyzed by astrobiologists in the 1990s. Here we have the famous worm image that was produced by scanning electron microscopy, and it was so tantalizing because these microscopic forms seemed to resemble fossils of microbial life. There was also complex organic material found in the form of polycyclic aromatic hydrocarbons, or PAHs. Carbonate globules in this meteorite were enriched in this heavy isotope of carbon, carbon-13, to an extent that on Earth could indicate biogenic activity. And fourth, there were these very peculiar magnetite crystals with distribution and shape similar to those produced by magnetotactic bacteria here on Earth. However, further study of this meteorite found that these microscopic forms are about an order of magnitude smaller than the smallest life forms known on Earth at that time, and they also have no internal structure that we could discern. That complex organic material, well, it turns out that by studying the cosmos, we found that these types of complex organic molecules litter the cosmos, and also just might be contamination from the fact that the meteorite spent the last 13,000 years on the surface of the Earth. The carbonate globules enriched in carbon-13 can also be formed abiotically, it's found, and those magnetite crystals could be formed by shock alteration of the meteorite. So, taking all of our knowledge and reevaluation of ALH 84001, let's see how that plays in the 5-question framework from before. So, have we detected an authentic signal? The answer to that is yes. Electron microscopy, the chemical analyses that were employed are pretty robust at detecting those signals, and the identification of those signals also is not really in question. These are quite well-established procedures for studying rocks on Earth and can be easily applied to rocks that come from Mars. So far, so good. But when you get to number 3, are there abiotic sources of the signal? As we have seen, that isotopic enrichment of carbon and also the magnetite grains can form abiotically. Are those expressions consistent with expressions of life? Well, some of the chemical expressions are, but those cell-like structures may be too small to be viable cells. And then finally, we know that terrestrial contamination always plays this confounding factor in material that has spent a long time on Earth. And so this is why today ALH 84001 is not widely considered to contain fossilized evidence of Martian life. So here we've seen how this standards of evidence approach can be put into action to help us understand whether or not we've found evidence of alien life in the cosmos. Okay, so with that brief recap of past biosignature assessment, let's now turn our gaze to the very exciting future. We are embarking on a golden age of solar system astrobiology in which numerous missions are being built and launched for the explicit purpose of trying to assess habitability and signs of life elsewhere in our solar system, in our cosmic backyard. We are in the saga of a Mars sample return mission. Europa Clipper launches in October. Dragonfly is going to visit Titan in the 2030s. And the latest decadal survey puts a very high recommendation on sending a flagship level mission to Enceladus to search for life in the icy plumes of those worlds. So now really is the time to try to develop cutting edge in situ biosignature techniques for solar system exploration, techniques that will hopefully be more definitive than any of the techniques tried before. So when I think about strong biosignature techniques, I think that they have four essential attributes. Number one is that they must be robust, which means immune to false positives and false negatives. Now, there may never be a sort of silver bullet biosignature technique that always gets it right, but we must try our best to make sure that our techniques are as immune as possible to false positives and false negatives. Second, a strong biosignature technique should be agnostic, which means capable of identifying life, even if that life does not share the exact same biochemistry as life on Earth. Third, the biosignature technique should be probabilistic, which means we must be able to quantify our confidence in the detection or non-detection of alien life. And finally, it's got to be flight ready. If the biosignature technique does not rely on instrumentation that NASA, ESA, or some other space agency is actually going to strap onto the back of a rocket and launch into space, then it's probably not going to be super useful in the near term. So Anirudh, in his portion of the talk, is going to talk about the robustness and the probabilistic nature of the technique that we're proposing. For now, I will focus on the agnostic nature and the flight readiness of the technique. Okay, so let's talk about agnostic biosignatures first. So to find truly alien life, or to widen our net so that we are capable of finding alien life that is not similar in detail, molecular detail, to life on Earth, we should develop these so-called agnostic biosignatures. So as you all know, a biosignature is a feature that is indicative or suggestive of the presence of life, either extant or extinct. And we tack on this word agnostic there to say that we are not trying to be specific to life as we know it on Earth, that our technique is capable of detecting even unfamiliar forms of life. And this is because there's a growing recognition in the astrobiology community that we cannot expect that life on other worlds with independent origins and different evolutionary paths will have the exact same details as life on Earth. So we kind of need to abstract away from certain details of chemistry here on Earth to be able to catch signatures of life that may be fundamental to life everywhere, but may not be instantiated in the exact substrates of life that we have here on Earth. So one way to do this, one common way of doing this, is appealing to some metric of complexity. Life is a quintessential complex evolving system, and complexity is maximized somewhere between total orderliness and utter chaos. And one might see life as a system in which information structures flows of matter and energy, and that structuring leads to some kind of complexity that can form the foundation for agnostic approaches to searching for life. Now, before diving into our own proposal for an agnostic biosignature, let me give you a few examples of agnostic biosignatures that have been developed by astrobiologists in the community in the recent years. So the first technique that I'll introduce you to is called chemometric fingerprinting. This was developed by Georgetown University professor Sarah Stewart Johnson and colleagues. The idea here is basically to use strands of DNA as different colored sticky notes, so to speak. The more complex your sample is, the more different kinds of DNA sequences you expect to bind to it. So in a simple sample, like an ordered crystal, that might only attract a few kinds of DNA molecules, but a very complex sample, like the heterogeneous nature of a cell membrane structured by proteins inside of it, might interact with a vast diversity of different DNA strands. So here we're basically measuring complexity through the diversity of DNA sequences that a sample interacts with. And for more information on this, please check out Sarah Stewart Johnson's 2018 paper published in Astrobiology. Another exciting technique that has been introduced to the community in recent years is called the molecular assembly. This was pioneered by Glasgow professor Lee Cronin and ASU professor Sarah Walker and their colleagues. And this basically asks how many unique transformations must one make to construct some object of interest, whether it's a series of building blocks or letters that form a word or chemical bonds that form an organic molecule. So using the assembly index, we're basically trying to measure the complexity of an organic molecule through the number of unique transformations that it takes to construct that molecule. I like to think of this as the Lego block principle. You know, if you go to the store and you buy a Lego set and the instruction booklet is super, super thin, you know it's a very easy set to make. But if the instruction booklet looks more like a dictionary, then you know you're going to be spending a lot of time and energy and information processing to make that final product. So the cool thing about assembly indices is that it seems like life is the only process capable of making really high molecular assembly index molecules. So I'm showing you 4 molecules here: ATP, penicillin, tryptophan, and asparagine. All 4 of these are used in life as we know it here on Earth, but life is the only process known to be able to create ATP and penicillin, whereas tryptophan and asparagine can be made abiotically through geochemical processes. So it seems like there is some cutoff roughly around assembly index 15, reported by Cronin, Walker, and colleagues, that basically above that, only life is responsible for making those complex molecules. Below that, it could be life or it could be an abiotic process too. And I encourage you all to visit the assembly theory papers, such as the Marshall et al. 2021 paper and the growing literature published by Cronin, Walker, and their group. So for us, assembly index, that's characterizing the complexity of an individual molecule. But the technique that Anirudh and I are working on is based on a complementary approach, dealing with molecular distributions as a whole within a sample that you might pick up on, say, the surface of Mars or in the plumes of Enceladus. And so the idea of using molecular distributions as a biosignature has been discussed for several decades. And the idea here is basically that we might expect living samples and non-living samples to have vastly different distributions of organic molecules. So in this graphic, you're seeing sort of an abiotic sample shown in that gray curve corresponding to maybe some thermodynamic distribution of organic molecules based on their molecular weight. But a biotic sample, which is shown in red, might have very, very specific molecular weights that are overrepresented in that sample. And now there's no guarantee that life on another world is going to use the exact same biomolecules as life on Earth, but we might expect that its distribution of organic molecules will be similarly different from the distribution of organic molecules in abiological samples, even while being different from the distribution of organic molecules in life on Earth. So why fundamentally is the case that the distribution of organic molecules in biotic samples should differ from those of abiotic samples? To try to answer that, I want to make an analogy to statistical mechanics. So, say you have a hot cup of tea that you have sitting on your desk. Now, the position and the momenta of all the teacup's molecules constitutes its microstate. And there are relatively few microstates that corresponds to a hot cup of tea in a room temperature room than correspond to a cold cup of tea in that room temperature room. And that's why this cup of tea will tend to cool down over time. This is the second law of thermodynamics. This is our familiar arrow of time. So the analogy that I want to make to life is this, that there may be some similar high dimensional phase space, not of position and momenta of particles, but of molecular patterns that differentiates life and non-life. So the curious thing, though, about life is that lifeless chemistry becomes self-organized into biochemistry. Non-living worlds turn into living worlds, sort of in the opposite direction of a warm cup cooling down. Seems to defy the second law of thermodynamics, but it doesn't. The key, of course, is that life is an open system, able to transfer and exchange matter, energy, and information with its environment, and that life also responds to selection pressures, in particular, selection for functions like homeostasis and resiliency and the ability to reproduce and learn about its environment, et cetera, et cetera. So we explore this idea of selection for function in another paper that we published last year titled On the Roles of Function and Selection in Evolving Systems. This was written by a group of scientists from a diverse range of fields, as well as philosophers of science who really helped us sharpen the ideas in this manuscript. I'm not going to say much about this paper, but I encourage you to go and check it out for more of our thoughts on the relationship between selection for function, evolution, and information processing. Okay, so back to biosignatures. The technique that Anirudh and I are going to introduce to you in just a second has its legacy in the work of Gutenberg et al. 2021, which was published in the journal Life. In this study, they used electrospray ionization Fourier transform ion cyclotron resonance mass spectrometry, or ESI-FT-ICRMS, to analyze the distribution of organic molecules in a diverse array of samples that that were alive, like E. coli, and samples that contain rich organic chemistry, but were never alive, such as the Murchison meteorite and the Miller-Urey experiment, one of those famous prebiotic chemistry experiments that can create amino acids, nucleobases, all sorts of sugar-like molecules in a prebiotic setting. And they trained a multi-class logistic regression classifier and achieved 90%, 92% accuracy in distinguishing one kind of sample from another. And this is really great. It's a wonderful proof of concept. The only catch is that it doesn't really satisfy that flight readiness. This electron ionization Fourier transform ion cyclotron resonance mass spectrometry is not just a very complicated term. It's also far too complicated of a technique to actually load onto a spacecraft and send to another world. So in this new work, we seek to rectify that. And here finally is the paper that Anirudh and I are really proud to present to you today. This is a robust agnostic biosignature based on machine learning and pyrolysis-GCMS. The lead author is our friend and colleague, Jim Cleaves. The team also includes Greta Heistad, a mathematician at Purdue University, George Cody, an organic chemist and astrobiologist at Carnegie Science, Sophia Economon, who is a graduate student in earth and planetary sciences at Johns Hopkins University, and Bob Hazen, who is a mineralogist and astrobiologist at Carnegie. So again, say you're tasked with trying to find life on another world, maybe Jupiter's moon Europa. How are you going to do it? You're going to send a probe, right? You're going to send a probe to analyze some of this, say, surface material, this plume detritus that probably originated in the habitable subsurface ocean below. And you might try to send one of these instruments to that world, a pyrolysis gas chromatograph mass spectrometer, or PY-GCMS. Now, if you're not familiar with what this instrument does, it's basically a very fancy oven that not only bakes your cake, but eats it for you too. So in detail, here's how a pyrolysis-GCMS works. So the first step is flash pyrolysis, followed by chromatographic separation, electron ionization, and then mass spectrometry using a quadruple mass analyzer. So the first step is to heat up a planetary sample very quickly in just over a second to 610 degrees Celsius and hold it for 10 seconds. This allows you to volatilize what we call pyrolysates, which can then enter into our gas chromatograph. The GC column is made up of phenyl polymethylsiloxane, and this material interacts with different chemicals differently. Some of the chemicals will be very speedy and go through the GC column very quickly and elute sooner than others, which interact with the column material differently in a more, say, sticky way. The GC oven increases in temperature at a rate of 5 degrees Celsius per minute from 50 Celsius to 300 Celsius, so it takes roughly an hour for one sample to work its way through the GC. After something comes out of the GC, it is ionized by 70 electron volts electron ionization. And then, of the things that come out of the GC at any given time, that may be composed of molecules with different masses. And once these molecules are ionized, they will respond to the quadrupole mass spectrometer differently. Some masses will just be thrown out of the mass spectrometer through an unstable trajectory, whereas others will make their way through and be detected by the detector in the mass spectrometer. And in this way, we're able to scan over all of the different masses and create a mass spectrum of every single bit of stuff that comes out of the GC at any given time. Okay, so there are several advantages to pyrolysis-GCMS that I want to highlight here. Number one is the rapid sample introduction and no sample prep related to flash pyrolysis. So you don't have to do any derivatization. You just need to take a little bit of sample, like a milligram or less of sample, and then just flash pyrolyze it. Second, gas chromatography is much simpler than methods that depend on mobile phases, such as liquid chromatography, which may not work so well in the very low temperature, low pressure conditions native to many planetary bodies of interest. And you also have this very low energy technique of ionization, 70 electron volts, which is good for spacecraft missions that may be energy limited. And then finally, the mass spectrum provides ample information about the chemistry of a sample. And we know this because we have shown that this as technique is proven on many spacecraft missions. In fact, we launch these GCMSs to interplanetary destinations all the time. As we speak, there's one sitting in the belly of the Curiosity rover on the surface of Mars. And so the data that ends up getting collected from a pyrolysis GCMS looks like this. On the x-axis, you have the mass to charge ratio of the various ions that are being counted through time as that sample chamber, as the GC heats up, right? So there's a time axis, there's a mass to charge axis, and then the z-axis is simply the intensity, the number of molecules that are being detected per second. And if you saw a distribution of organic molecules that looked like this on some alien world, I think you'd be very excited because this is what the average Earth bacterium looks like. And what we've been doing at Carnegie is taking all sorts of very diverse organic rich samples to build up our training set to train a machine learning model that Anirudh will speak about in just a second. We've got organic matter that was very recently alive, like bacteria and plants, living matter that has been geologically degraded over millions of years. It was once alive, but now it's, petroleum or coal or oil or something like that. And then, of course, we have a bunch of organic rich materials that were never alive, carbonaceous chondrites, etc. And so the question is, can we quantify how these distributions of organic molecules are similar to and different from one another? And can we train a machine learning algorithm to tell the difference between something that was once alive and something that was never alive? And it turns out very excitingly, the answer to that is yes. And I will turn it over to Anirudh Prabhu to tell you how.
Thanks, Mike. My turn to share my screen. So let's see how this goes. Can everyone see my screen? Just give me some kind of a thumbs up if you can.
You're good.
Okay, sounds good. All right, so thanks, Mike. That's a wonderful introduction to the problem and to me, so I'm not going to spend too much time on an initial slide. Hello, everyone. My name is Anirudh Prabhu. I'm a research scientist and PI at Carnegie Institute for Science, and today I'll be taking you through the machine learning component of one of our recent big advances, right? So to start off, as Mike mentioned, we'll rehydrate the point. A good biosignature detection method should meet four main criteria, right? It should be agnostic. That is, it should be capable of identifying signs of life different from Earth's biology. It should be probabilistic. The model or method that should be developed should be able to quantify the confidence in the biosignature that it has detected. It should be robust. The method should be consistent and accurate at detecting said biosignatures. And of course, Mike spent a lot of time talking about why it's important for this kind of method to be flight ready. As I go through this talk, I'll talk through especially how our method is probabilistic and robust, and also show you an example of the agnosticness that Mike talked about. So to start off, as has been repeatedly said, we used pyrolysis-GCMS to analyze about 134 samples. We analyzed a good mix of biotic and abiotic samples, including leaves, fossils, meteorites, nylon, etc. And as Mike already pointed out, this is what our data looks like. The data produced by the PyGCMS has three main attributes, right? We have rows represented by scan numbers, which is a proxy for the retention time. We have columns representing mass to charge ratios. We have intensity values for each row and column combination. So most of the time, researchers that use PyGCMS, they look at this data, they identify certain peaks in the data, and use those peaks to identify specific molecules. Oh, wait, do you not see the slides changing?
I do see the slides changing.
Okay,
So
I just got a comment, so I just wanted to make sure y'all are following along and, okay, thanks. So these identified molecules are then used by the researchers to ascertain the nature of the sample, whether it is biotic or abiotic. So let's see. So most researchers that use GCMS, this is the normal mode of operations, right? So we, on the other hand, look at the entire data set. As a data scientist, I always like looking at the entire data set, but in this case specifically, we look at the positions of the peaks, the intensity values, and the relationships between the peaks that are at various positions. So for example, you can see in this figure, the aggregated GCMS data for bacteria, for plants, for petroleum samples, and for meteorites, right? And you see each of them looks significantly different from each other. So what we're trying to do here is look at the entire landscape and use our model to then find recurring patterns in the data and use those patterns to predict the biogenicity of the sample. So what you see here is an ultra simplified workflow of our data, which we started with running the GCMS on 134 samples. The next step was pre-processing the data to get it ready for the machine learning models. So we tried quite a few pre-processing strategies and sort of stitched this method together, but the most common steps for us included smoothing the mass spec data, baseline removal, normalizing the intensity for each of the mass specs, um, and peak detection. Lastly, we flattened our data set from the 134 samples from a 3D object to sort of a 2D object to make it ready for machine learning. And lastly, we binned the data to make it more computationally efficient. So once the pre-processing was done, we could get to the crux of our approach, which was building and testing many different classifiers that could predict the biotic or abiotic nature of a sample. So we tried quite a few classifiers. We tried support vector machines, extreme gradient boosting. We tried different neural network methods. Um, what we found was that random forest was the model that performed the best. I'll go into detail in upcoming slides, but a short introduction of random forest is that it's a very popular nonlinear model that aggregates over many decision trees to improve its predictive capabilities. The reason I just gave you a short intro before going into detail later is because I was really excited to tell you guys that, as we were testing the random forest model and improving its generalizability, we found that our first iteration, the one that we published, the random forest model, can distinguish biotic and abiotic samples with close to 90% accuracy. So we were so excited with that, right? So now to actually explain the random forest method and also sort of give you a clue of why it does so well and why I particularly like to use it over some of the more popular neural network-based methods or deep learning-based methods. So random forest method works as follows. We take a random subset of our data and use it to build a decision tree. A decision tree is something most of you are familiar with. It starts with looking at the features of the data and trying to make decisions based on what it is you're trying to predict, which in this case is a classifier that will assign whether a sample is biotic or abiotic. So what we do in a random forest is we take many such random subsets of our data. We do it many, many times. In our case, we did it about 500 times. You can do it thousands of times if you wish, if the model keeps improving in each iteration. And we build a slightly different decision tree each time, right? So each time you take a different random subset and try to say, based on this, what is the path to a sample being biotic or abiotic? And now with many such decision trees, you have a forest. So hence random forest. So now that we have a decision forest, we can predict the biogenicity of an unknown sample. So what happens when we have a new sample to be tested using our model? We put that sample through every single decision tree. Every decision tree then gets a vote. So it'll vote whether, according to that decision tree, the model is biotic or abiotic. And we aggregate the results of the entire forest to form our final prediction. Now, because each tree was made from a random subset of samples, each tree looks at a different view of the data. So to continue Mike's analogy, right? If we had to slice this cake from very many different angles. If we had to make so many different slices of our data, not every slice might have all the ingredients of the cake. So we want to avoid ending up with a biased view of our data, and we want a generalized model. So we aggregate the results of 500 or more decision trees to make our final prediction. And we use an out of bag error, which is whatever is left from those random subsetting of samples that we made. Those test samples are then used to calculate what we call an out of bag error. And that is the prediction accuracy of the model, or the first instance of prediction accuracy of the model that we get. And that is what we try to improve over iterations using feature selection and by fine-tuning our hyperparameters. So now that we've divided our model up, we've ensured the out-of-bag error is optimized. How do we ensure that our model is robust and accurate, right? We started with a fairly common process in machine learning. We divided our data into training and test data with a 70-30 split. That 30% of the test data that we had, we set it aside to never be introduced to the model at all, right? We don't want that test data to be touched by the model in any kind of way. Then with the 70% of the data, we ran nested cross validation with, I think, 5-fold inner loop and 10-fold outer loop. Or maybe I'm switching those around. But we ran a nested cross validation to make sure that our model was robustly predicting results each time. And coincidentally, we also found that the nested cross validation also showed close to a 90% accuracy. We still weren't satisfied that this is going to work in a generalized manner. So on top of this, we ran this nested cross validation many times using a Monte Carlo simulation method where we randomly sampled the data and changed our data set, and to make sure that the common sort of accuracy of our model still stayed around 90%. So once this was done, we finally tested the 30% of the untouched data and found that in our untouched data, the model was able to predict 90% of the occurrences of our samples correctly. Whether it was biotic or abiotic, it was able to figure this out correctly, which is great. So now that we have our first set of very promising results and this model can be applied, we then became very interested in actually interpreting our model and interpreting our results. So what you see here is a fairly common PCA plot. This plot shows us how similar or dissimilar the analyzed samples are. So this is one of the parallel approaches we did to our random forest model, just to see our interpretations of the model that it generated. So you can see here, the first two principal components account for about 55% of the variance in the data. So the other 18 plus components that we had show the other 45% of the variance of the data. This really tells you how complex the data is that we are analyzing. So we colored these samples by their biotic and abiotic nature. We did so at least initially until we noticed that there was a clear bifurcation in the biotic samples that were spreading to the right. This is not something we trained the model to identify. It's just something that the model identified in the molecular distributions of the data. So then we were like, well, this is a new result. We don't know why it's doing this, but it's really cool that it can bifurcate biotic samples into two types. And as we examined it, we found that, of course, the abiotic samples in orange are clumped to the bottom left in one tight cluster. But the biotic samples that start from the left and spread to the right in two different directions actually corresponded to fresh and degraded biotic samples. So in spite of us not telling the algorithm to learn these types of biotic samples, it was able to show this distinction between the two samples. Another point I'd like to highlight here, bring back full circle, is for why I think our model is agnostic. So let's look at this scenario as a test case, right? Let's say you have 3 unknown samples that you want to test the biogenicity for using our method. So you can see, based on the molecular distributions produced by the sample, they'll assume their place somewhere on this PCA plot. So in this example, you can see that unknown samples 1 and 3 squarely place themselves between existing samples, right? And if I had to venture a guess, sample 3 will probably be predicted as a fresh biotic sample. But what about sample 2, right? Sample 2 is up to the top left. It's behaving unlike any of the biotic or abiotic samples that we've trained on so far. So this could mean one of two things, right? The first very obvious thing it could mean is that our training set can and should be expanded. We are very much in the process of doing that right now. We have about 200 to 250 samples that we are actively running, and we're expanding our training set with various kinds of biotic and abiotic samples to make sure we have a larger, more comprehensive data set. But the other more exciting possibility, right, is that that particular sample, unknown sample two's molecular distributions are unlike any of the biotic or abiotic samples on Earth. Now, that's a very exciting thought. And we've now been able to identify this potentially anomalous sample. So we've identified an anomaly which is unlike the biotic or abiotic samples on Earth. And we've certainly narrowed down which samples should be studied for finding life or potential life on other planets different from what we know on our own. So that's pretty cool. So to summarize, we've discovered a potentially agnostic method to distinguish between biotic and abiotic samples. This distinction is not based on the identity of the molecules, but rather on the molecular distributions themselves, which may point to a fundamental difference between abiotic organic mixtures and biotic molecular suits. Lastly, our method is extendable, so it can be applied to analyze many different samples, including those from GCMS instruments that are used on board existing space missions or future space missions. And it generally looks for this kind of 3D spectral data, and this should be extendable beyond even GCMS potentially. So with the takeaways done, I want to go through the 4 criteria for a good biosignature detection method again. I've already told you about an example of how our method can identify anomalies and thus is agnostic. But going over the other key points, a machine learning method like random forest can certainly provide a quantified confidence value in each prediction that it makes, thus making our method probabilistic. And here's where I'll point out why I like random forest just a little bit better, because if you can examine some of the decision trees and look at the repeating features that lead to the predictions and see how important each feature is, then it's less black boxy compared to some other machine learning methods, and you can inquire into the data and gain a better explanation for why a particular sample may be biotic. With our steps to assess generalizability using nested cross-validation, feature selection, and our model still maintaining close to a 90% accuracy, I'm going to consider our method robust at detecting biosignatures. And Mike has already spent a lot of time talking about this, but it's really important that this method is flight ready. And with the GCMS instrument being flight ready, this could just be a method that works with that data and detects biosignatures. So with all that said and some results and the method presented, what do we have planned next for this project? Well, there are 2 big directions that we are pursuing for this project right now: an astrobiological path forward and a paleobiological one. On the astrobiological side, we have an opportunity to detect life on other planets when our method is combined with a flight-ready instrument like the GCMS. Another ongoing exploration of ours is studying the data produced by a mixture of the biotic and abiotic samples. Now, this is done to simulate real samples we may find on other planets. So if there's a rover on another planet, it's going to be pretty hard to clean the sample or to process it in any way. And to simulate and test that, our intern, Emmy Slaughter, has spent the past month running a mixture of biotic and abiotic samples at various ratios. And we're testing if we can see and detect the biogenicity of the sample when it's mixed with a bunch of abiotic material. And really, we're pushing the boundaries to see exactly how much abiotic mixture can be in a biotic sample for the model to still accurately recognize the biotic sample. Now, on the paleobiological side, we plan to develop a more granular classifier to characterize the deep time samples. We've already received about 200 deep time samples from 25 of the world's leading paleobiologists. We hope that our work in this side will help improve our understanding of early life, and some of Earth's oldest and enigmatic samples like apex shirt can be studied, and we may be able to look at their true nature. Now, for the paleobiological exploration, a lot of you might be wondering how it's possible to get information from such a degraded old sample. So if you've been looking at science news recently, you would have seen articles about a student who identified and translated an extremely old and degraded Roman scroll that was carbonized by Mount Vesuvius's eruption in 79 CE. So the machine learning method that the student used was able to identify characters from a very degraded scroll when there was very little experts could actually read and gain from that scroll. So this is similar to how we're thinking about these paleobiological samples, right? In spite of the degradation in these very old samples, there's still some information left in the molecular distributions to make predictions about it. And we hope our method can be used to study such samples. That's it from me. Thank you.
Guys, that was fascinating. So, okay, perfect. I see. So if you have questions, please use the raise hand option or put them in the chat. And I actually see, it looks like we have a question right away from Ruben. Are either of you able to see it? If not, I can also read it. Actually, I'm just going to read it to you guys. As Tellurian life usually favors one chirality over the other for each macromolecule, do you have the capacity to test for left and right-handed molecules? And if so, how do you make sure to train your AI to account for alien combinations of chiralities?
I think I'll take this one. So, hey, Ruben, great question. Yeah, I don't think our instrument is able to tell the difference between left and right-handed organic molecules. So my hope is that when employed, our methodology will be one of many instruments and techniques that will be applied to extraterrestrial samples in a sort of perhaps even Bayesian framework of trying to integrate a whole bunch of independent lines of evidence to try to distinguish life from non-life on different worlds. So definitely chirality is something that you'd want to look at, and hopefully in combination with that and other kinds of measurements that you'll make, our technique will be added to that, and we'll be able to use all of these things in combination, right? Because multiple lines of evidence are essential for astrobiology and biosignature detection. So that's a great question.
And let's go to people with their hands raised. Mary, it looks like you're number one. So Mary Wojtek, go for it.
I am number one. Thank you, Ryan. I just want to say this was a fantastic talk. Thank you, guys. And I really love your technique. I love that it's flight ready. I love that there's the probabilistic aspect of it. And I love actually your direction to do a mixture of samples, because I think that's going to make a huge difference. I think a lot of the biosignatures we have, once you start mixing it with non-biological samples, it becomes a nightmare. And just to respond a little bit to the chirality question, we now know that abiotic samples can be homochiral as well, because it's just a feature of reactions and reaction kinetics and efficiency. But so after saying you guys are great, my question is, I know that the two other techniques that you mentioned, and the one I know them are have the potential for false negatives. Something is you either have a DNA strand or you don't, if it's some kind of other kind of life. And with the molecular assembly, it's at a threshold where, you're 100% certain that it's biological, but there's so much that can be biological that won't be detected. So my question for you is, with a 90% certainty, have you thought about what other kind of measurements you would like to have that would help you? Have you thought about how to reduce that? And have you thought of, or I guess, increase the confidence, not reduce your confidence? And what other measurements you'd do complementary to actually increase the confidence? So long
Right.
question, but thank you.
Okay, from the I'll do the sort of the instrument and data gathering side, and then Anirudh, you can talk about more of the machine learning stuff. So obviously, so the first paper, the one that was published last year, used 134 different samples, and right now we have roughly 300 additional samples that we're going to fold into this process. So hopefully, with a vastly greater data set on which to train our machine learning method, we will be able to increase the confidence. And if we can also categorize our samples in a more fine-grained way instead of just biotic or abiotic, but perhaps like meteorites, synthetic materials, bacterial cells, eukaryotes, that kind of distinctions will also potentially help us craft an even greater confidence in accuracy in our findings. And so yeah, we're just working on expanding the training set right now, and then playing around with further categorization.
Yeah, I'll take part two, just add to Mike's answer, right? I'm obviously going to start off with the generic data science answer as well. The more data, the better, right? So the more data we gain. So Mike talked about one aspect of that data, which was, training more samples and testing different samples at different conditions and producing this data. But depending on the kind of flight instrument that is there, combined with the PyGCMS, we were also thinking about maybe analyzing the same samples through other data to produce sort of a layered data set for every sample. So looking at Raman maybe, or looking at some other instruments that are able to tell us other aspects, right, of that sample that the PyGCMS cannot. And then adding this as a layer to like make a thicker sort of book for that individual sample. And so instead of even training just more samples, if we can get more information about the same sample, they can be layered into this machine learning method or other machine learning methods that we're going to test out. And that I think may lead to a better prediction. My goal is when we do this to maintain at least our current predictions, right? When you have more, like the confidence goes down. So in the next iteration, if we're able to add this data and keep the 90%, I'm very satisfied. And then we're going to step up to reduce the error a bit more.
Thank you.
Thanks, Mary.
Okay, we're going to hop back over to chat real quick. We have a lot of questions, and also just I put this in chat since we're coming close to the hour. If you do have your hand raised, please feel free to post your question in the chat, and I will give Mike and Anirudh an opportunity to respond to you outside of the seminar. They can always email you or find you somewhere else. So we're going to go back to Slack real quick. Does changing external environment conditions affect the data? If yes, and how do you account for that in testing and training data?
Yeah, so that's a good question. I assume you mean like if you have this instrument operating on a different world and maybe that world is experiencing diurnal cycles or even seasonal cycles or a dust storm or whatever, how does that affect things? And yes, so the idea is that we're relying on an instrument that is able to keep itself relatively stable in terms of, the temperature ramp in the GC, for instance, is consistent with each run. If it's not, then that's going to be a problem, but that would be a problem for basically any kind of instrument. If that instrument isn't insulated from the environmental changes, it will produce potentially garbage, right? So we are relying on the ability for the instrument to maintain some kind of optimal state. And right now, the instrument that we're generating our data set in is securely housed in a well-regulated laboratory with constant temperature and humidity and things like that we're monitoring. But yeah, we'd have to contend with potentially, how to protect this instrument from environmental fluctuations on another world. But I think that's been not too big of an issue if I've been following correctly some of the MSL things and things like that. But again, yeah, obviously something that the engineers will have to think about as we prepare to potentially launch our instrument and our methodology to extraterrestrial destinations.
Okay. Jessica, I see you've got your hand raised. Go for it.
Oh, that was a mistake. Sorry.
Oh, okay. Nashkia, you have your hand raised. Go for it.
Yes, I do. I was actually just typing my question, but I'm just gonna read that now. Okay. So thanks also for this really exciting and very clear talk. I learned a lot. I have two questions. The first one is just to clarify the plot that you had, Anirudh, on the PC1, PC2. What were maybe you explained it or maybe I missed it, but what is exactly the shaded regions? Like what is the shaded kind of boundaries and what does that mean and why do they have those shapes? And then yeah, and the second one, so maybe you want to explain that first and then I'll because
Yeah,
it's
sure.
simple.
So the shaded region was purely representational, right? So that's a good question. Uh, the PCA plot just showed how similar or dissimilar, the different samples are, and we then colored initially just those points by whether we knew whether they were biotic and abiotic. Uh, we really wanted to highlight the original diagram doesn't have any shades. We haven't run any clustering on it yet, or at least anything that's published yet on those results. So those boundaries are purely Mike drew them up because it's easier to tell the groups
Oh,
apart.
okay.
And so there's no data-driven boundaries there at this stage. Good.
Okay. So it was just by eye, basically. You just did that. Okay. Okay, cool. And the second question is, okay, one of the criteria that Mike was explaining about, that you need independent lines of evidence to detect life. So for me, in your work, it means that detecting life should be method independent, right? And I really like that you use random forest because, as you said, it's more controlled and you're able to see what features are being selected and then it's much easier to know what features are going to be important, right? So if this if the answers that you're getting should be method independent, have you compared it with other more other neural networks and other methods, and are you getting the same answer? And to what extent should you be expecting to get the same answer, given that, I guess other methods have it's more complicated to know which are the important features in those methods.
Right. So again, great question. We have got similar results with varying degrees of accuracy in other models, right? For the first iteration that we ran, I mentioned that we ran support vector machines, neural networks, and extreme gradient boosting. And XGBoost came close to the accuracy that the random forest had, but others were lower. I'm not going to say significantly lower, but they were lower. And so we went with the best model. But I do believe our results are, in terms of the confusion matrices that were generated from each of those, are available as part of the supplementary material. And so we'll know how good each method was at identifying certain samples. And that we were discussing and we were planning to publish this in a more detailed data set paper analyzing these methods, which our colleague Greta Heisted is working on what methods are good at identifying what type of samples, right? And there is a difference in how these methods work, which is an interesting finding. So while these are data agnostic, if you have a similar kind of data, these methods should work, irrespective of the type of sample they are. They aren't quite method agnostic just yet. And that may be a product of a combination of things, including how we pre-process our data. So we're really trying to try all the different methods with all the different pre-processing methods that we have to see how we can generalize and standardize this. And we publish the best thing first.
Right.